Dr. Michael Dominique Roberts.
Dominic.
Put a little Tennessee on there. Buddy, we're here.
Here we are.
I'm excited.
Me too.
To welcome you and our listeners to the very first Fitomics podcast at our headquarters in Alabaster, Alabama. We're also joined by Dr. Daniel Lawson.
Hello.
And at Master Control is our esteemed Chief Medical Officer, my good friend David Michael Hardin, medical doctor. So today we want to talk about a variety of things, but particularly muscle.
Mm-hmm.
Muscle. Before diving right into that, I wanted you to have an opportunity to share about the Molecular and Applied Sciences Lab at Auburn.
Okay.
Which I was fortunate. Now you are still probably regretting your decision to do my PhD under your tutelage, man, and just incredibly grateful for that time.
Yeah.
I think a big reason that I'm sitting here today is because of that.
Oh, you're too
and
kind.
Everything you taught me, and I appreciate your patience as I likely annoyed you regularly. And now being out of it and, being a business owner, and understanding the financial implications of decisions when it comes to R&D emphasis and interests, like you were always, I can't think of a time that you didn't, you kept your door cracked, which was cool. I always felt like I was able to come in and, Doc, can we do this? Like, buddy, the antibodies are X hundreds of dollars. And yeah, I mean, it's interesting, but we just gotta, but you were always like just super kind, man. And I appreciate that very much.
Thank you.
I was saying before, we started that when I read Teddy Roosevelt's Man in the Arena speech, I think of you.
I appreciate that.
Like you're about it. You're about the science. You are a person of high integrity and character. I'll stop rambling, but I love you, man. I'm glad you're here.
I love you too. And this is quite impressive. And thank you. I feel incredibly honored to be y'all's first podcast guest, which I'm sure will turn into several hundred podcasts. So this is quite an honor. But yeah, so I am a researcher at Auburn University, and I direct what is now named the Nutrabolt lab. So we can get into that in a little bit. But that was made possible through the kind generosity of one of our common friends, Chris Lockwood.
Oh, yeah. Shout out to Chris Lockwood, man.
Yes. Also a great content guy as it relates to nutritional supplements, the regulatory framework, et cetera. But Chris understands the financial challenges of research, and he just wanted to provide a 3-year lab gift, really with no strings attached. And it was us that said, "Hey, why don't we tag y'all's company's name to the lab?" But the intent is to provide funds, resources, liquidity, so that students who are like you, just intellectually curious, can run projects, get the reagents, travel to conferences, things like that. So
that's cool, man.
Yeah, huge shout out to Chris. And
that's amazing.
I tell people, there's always a stigma with doing or being engaged in research, in particular with industry. I've never really understood why. I think the academic model, and again, this is something we'll probably talk about during the podcast, but they're conditioned to chase federal dollars, NIH money.
Where do those come from?
So they are probably, that money is probably printed. But anyways, so, with industry, right? I mean, they take some of their profits, they set it aside, and they either donate through philanthropy or, they have a product that they believe in, and they truly want to see if it works in the context of performance enhancement, et cetera. So huge respect for our industry partnerships and what you all are doing here at Fitomics. I really believe that there needs to be more academic cross-pollination with these endeavors.
Yeah, man. I mean, the research, I like to define it as the search for truth.
Mm-hmm.
And whether you're in academia or in industry, I hope that's what we're doing. And the marketing is honest.
Yeah.
And the product is well formulated. And, the claims are evidentially true,
Sure.
not just made up.
Yep.
And yeah, Chris is solid. He's as long as I've known him, been about that.
100%.
So that's cool, man.
Yeah.
So that'll give some runway on some cool experiments.
It will.
Do you
Yeah.
want to talk about like your current studies like now and kind of
Sure.
next few
Yeah.
years with
Yeah.
that?
So in the context of just generalities and what we do at Auburn, we have a heavy emphasis on how exercise and more recently aging affect muscle biology. And so some of the projects we do can be as simple as getting a cohort of younger individuals, older individuals, uh, performing muscle imaging techniques, such as ultrasound, MRI, DEXA, not really muscle imaging, but you can get lean tissue mass outcomes. We can also get muscle biopsies. So I've taken 2,200. You took like 30 of them during your
It
time
was about
there.
100.
Oh yeah. So, and at once you get the biopsy, I mean the world opens up because you can do things as simple as Western blotting to look at single protein targets or qPCR for single mRNA expression targets, we can send it to collaborators and look at the epigenetic profile, how it differs between younger and older individuals. We can look at the proteomic profile and get the entire plethora of proteins in the muscle to see how aging affects that landscape. We can do transcriptomics recently, and I'll discuss a study we're doing with Max Michel in the laboratory, but we were getting into small RNA sequencing to look at microRNAs and non-coding RNAs and things like that. So, an example of a project would be cohort of older people, cohort of younger people. Let's do some of our non-invasive techniques for muscle imaging. Let's get a biopsy. Let's interrogate it at the molecular level, and let's tell the story. Right? How does aging affect the proteomic landscape? What are the implications? We can also do studies where we take older people, we have them resistance train anywhere between 8 to 16 weeks, and we can see what happens in that tissue at the molecular level, right? So, yeah, we've been a lot more interested in aging because I think I heard Stu Phillips say at some point research becomes me search, and I'm aging. So, I'm looking to try to stay as young as possible. And we clearly know exercise can do that. So I am interested more so now than ever in how we can leverage resistance training to rejuvenate the muscle. And we have great data on that. Well, I think on the phone you and I were talking about certain small molecules like NAD. We published a study recently showing that in an older cohort of individuals, right, you can drastically increase that metabolite muscle through resistance training. We have some data showing that at the epigenetic level, when older individuals resistance train, we can actually cause methylation changes to the mitochondrial genome, which then should trigger mitochondrial biogenesis. And yeah, so that's sort of generally what we do. We also do nutritional supplement related research. We did a study with Nutrabolt that was not tied to the gift, but we were looking at one of their healthy hydration drinks, showing it reduced cramps, and after or taken before a long treadmill bout. And we've done a lot of studies. I mean, when you were at Auburn, we compared the effects of whey protein versus soy protein versus leucine only. That was in collaboration with Chris Lockwood. Um, showing that practically speaking, if you start resistance training, it really isn't the supplementation that matters so much. It's the consistency. And as you become more dialed in and more consistent, then you can start thinking about working on the fringes with certain supplements. But yeah, been doing it for 13 years at Auburn. And what the future, I think you'll see a little bit more aging related research questions, but I'm also interested, frankly, in what you all do here, because I do think there is a disconnect between the sterile lab environment and what we measure versus what people come to you all for. And so, I've been interested in different training methodologies. I have a very gifted student, Danny Plotkin, right now. He's very in tune with social media as well as the scientific literature. And he wants to compare length and contractions, training that way for, an 8-week period versus conventional range of motion resistance training, and whether or not you get enhanced hypertrophy with that length and contraction modality. And that's been done, and there's good data on that. But I think what we're going to add to that body of literature is we took the biopsies, right? We're going to see.
Broke the skin.
We broke the skin, which is hard to do.
Mm-hmm.
Right.
Yeah. It's an involved, basically outpatient surgery.
It is. And it's, I've done, like I said, 2,200. This is a bit of an aside. We've only had one issue where we had somebody bleeding. It was a little subskin blood vessel. And so, we had to stitch them. It's not terribly traumatic. We have people doing max testing sometimes afterwards.
Right.
But, I'm a dad of 4, right? Every time I am inflicting that sort of sensation on somebody, I mean, you lose sleep. You just hope that they're okay. And when they come back in the next day, like, yeah, I'm fine. You're like, okay, thank God. I don't want to hurt people. So, anyways, now I'm rambling, but that's kind of the gist. Is that we really take pride, and you were a very large part of that. I mean, you really started this golden era in our laboratory with the hypertrophy related research, and now we have guys like Danny Plotkin, Brad Rupel, who's postdocing right now, Josh Godwin. They carried that torch, and they did a lot of really cool, elegant studies. So, which I hope we talk about today.
Oh man, we're gonna dive in. I think that was a great summary, man. Oh man, how many labs would you say in the States in the exercise science space are regularly collecting muscle biopsies? And just to give some context, right?
Yeah.
How we have learned about bioenergetics and we're talking omics, the protein pool and RNA and satellite cells, all these things about the physiology of muscle.
Yeah.
Got to take biopsies.
Yeah.
So it's people like you and Booth and well, he mainly did rodent models.
Sure. But that important point, because I've seen this come across certain organizations, social media. Who cares? It's a mouse. But there is certainly really cool things you can do in mice, right? Troy Hornberger comes to mind. He just set up a model where it is kind of like a weighted sled pull, progressive overload, showing similar
mice jacked.
Yes.
Yeah.
And similar hypertrophy on a percentage basis when you compare it to human training studies. And so using proper manipulation, I think when doing these rodent models can lead to really cool findings. And what you can do with the mouse in particular is you can genetically manipulate it, right? So John McCarthy with Karen Esser back in 2010, they generated a mouse model where when you put a certain chemical in the water, it kills off the satellite cells. And that led to a body of literature where they were interested in saying, do we need satellite cells to help facilitate that overload induced muscle hypertrophy? The answer is yes, you do. And, it wasn't at the time, 2011, first study came out, sort of mixed findings, but that group, along with other researchers, published, I would say, over a dozen studies now indicating that, yes, satellite cells are important for muscle hypertrophy. And then with our human research, we take it a step further. We say, not only is it important, But when you look at like a higher hypertrophic responder, in fact, you see that they have this increased satellite cell activity. We found that. Marcus Bauman's found that. Bauman actually published the first study back in 2005 showing that. So yeah, there is a place for this basic preclinical mouse model research, and if you marry that with well-designed human studies, you can get a more complete picture.
Right, right. Yeah. And zooming out, I mean, that's really what part of our mission is to help bridge that gap.
Yeah.
Right. Like, again, searching for truth through research, but we want to bring that information to people to help them train more effectively, eat more effectively, be healthier for longer.
Yeah.
And, our conviction is research in that process of basic to taking it to the human model.
Sure.
That's and I think Boothright he discovered AMPK in a rodent model as far as its relationship with mitochondrial biogenesis.
So he trained with John Halsey, and John Halsey, that laboratory at WashU in the mid 1960s showed that when you exercise, this process of mitochondrial biogenesis occurs in skeletal muscle. And then Booth carried the torch, and he began his independent research career at Texas, showing that if we have a period of disuse, we have this exponential decay in muscle mitochondria. But if it wasn't for that Halsey lab and then Booth carrying the torch, and they had others like Mike Rennie was in that lab, Will Winder was in that lab, Ron Young, all the way to Keith Barr trained with John Halsey,
Yeah.
right? So Halsey really was kind of that grandfather of exercise muscle biology. Booth, in particular, he really became the godfather of molecular muscle. And I had postdoc with Frank for that very reason, because he was the first to do transcriptomics with muscle. He was the first, him and Pete Watson in the 1980s, they were the first to measure messenger RNA in muscle, right, using northern blotting.
In a rodent model.
In a rodent model. But no one prior to that point had measured messenger RNA. Frank was actually the first to use, it wasn't AAV, I think it was a lentivirus, but gene therapy in muscle, right? So Frank really was at the forefront of molecular, and having read his papers as a PhD student, I think I was maybe 2 years into a 4-year program. I said, I either want to go there with that guy or Karen Esser, right? So Karen is a legend. I mean, she trained Troy Hornberger, Gustavo Nader, Keith Barr, John McCarthy was a research assistant professor. So when we talk about the molecular response to muscle hypertrophy, we talk about ribosome biogenesis, right? Nader and Esser published that first paper. That was an in vitro paper. Karen and Keith, right, 1999, they were the first to show that mTORC1 signaling was operative in response to mechanical overload. So, I mean, just things we take for granted now. And you can do a quick Google search or go to Anthropic and use AI to say what happens in muscle with, these are the people that laid down the initial foundation.
In the arena.
That's right. Yeah.
And I guess just driving the point home that a lot of that work was done either in culture or in rodents, and
Amen.
we've taken that to the humans.
Yes.
And then, that's helped us to inform exercise prescription and
100%.
and
Yes.
so on.
Yeah. So I'm curious how, if you had to put like a percentage on it, how much does a mouse or rodent model translate to a human model?
Man. So I think in relation to exercise adaptation, there is a very high translation. So as an example, and we can go to Karen, piece of paper, or Subodine when she was with David Glass, right? Back in the late 90s, early 2000s, when they were doing synergist ablation. Now, the model of synergist ablation we can get into, but that's where you remove basically the gastroc from the rodent, and the intact muscles, once you sew the leg up, will then hypertrophy dramatically, right? So not terribly human relevant in terms of, humans being able to increase a given muscle's mass by, we'll call it 50 to 100%.
But it is compensatory overload.
It's
Like
compensatory overload. And in addition to that, the mechanisms that are operative are highly translatable when you look at human weight training studies. So mTORC1, Karen, Subodine, they pretty much figured that out. I think it was 6 or 7 years later when Blake Rasmussen's lab sort of replicated those findings. Schiaffino over in Italy, he showed that with compensatory hypertrophy, you have these little cells that reside in the extracellular matrix that proliferate. We now know them as satellite cells. And again, using the model showing that, right? So I think muscle biology in the context of exercise, and then working back to Frank and John Halsey and the mitochondrial biogenesis, we know that happens when you take biopsies out of humans when they do endurance training. I think there is a high translatability in regard to the exercise stressor on the muscle, whether it's a rodent or a human. But now, right, if you talk about cancer therapeutics, I teach a research methods course at Auburn, and if we talk about drugs that lower cholesterol, cancer therapeutics, et cetera, yeah, the hit rate's not that high. It seems to be somewhere between 30 and 40% when you look at the rodent data and then what actually works in a human. Not my area, so I can't intimately speak as to why that is. But we do know that there are metabolic differences in a mouse, for instance, versus a rat, even, and from rodents to humans. So that could explain some of that. But yeah, to answer the question directly, I mean, the muscle response seems to be highly conserved between mammalian species.
Yeah. So rodent research to understand muscle biology, muscle physiology isn't a waste of time, we could say.
Not at all. Yeah.
Well, probably a good opportunity to transition to why does this matter? Like as far as growing muscle.
Sure.
Muscle mass, muscle strength, muscle power. I really have enjoyed actually in the last few years. I think Gabrielle Lyon, the MD, she has promoted this idea of muscle being the organ of longevity, and how being under muscled in some senses perhaps worse for an individual than being over fat even. And sometimes, they go together.
Coincide, yeah.
Skeletal muscle. Why does it matter for our health, for our longevity, beyond just how absolutely awesome it is to get a gnarly bicep pump?
Yeah. This obviously gets somewhat philosophical and not grounded in hard literature, but as a species, right? We needed muscle when we didn't have grocery stores because we had to ambulate to forage for food. We had to sprint to survive, right? We had to climb things. We had to have fine dexterity using muscles to make tools. So, in that sense, muscle mattered back then. Now,
so we had to survive.
Muscle allowed us to survive. Central control tells muscles what to do in order to survive. Nowadays, we have grocery stores, right? A lot of sedentary time. So at this point, muscle is healthy. Healthy muscle is good for metabolism. If you exercise, you're going to have muscle cells utilizing substrate so that they don't accumulate in the liver or even in the muscle as fat, right? So in the current day sense, maintaining muscle is important. Healthy muscle is important because it keeps us metabolically healthy. And then in addition to that, you think about being able to perform daily activities of independent living. We need to maintain a certain critical threshold of strength, of power, of mass in order to do those things. And so that's why muscle is important. Now, the reason I got into muscle biology to begin with was my fascination with physique, right? I was an avid resistance trainer. I went to Baylor University. I was a biology major, but I loved resistance training. I loved taking certain supplements. And I was always fascinated with the Ronnie Colemans, the Flex Wheelers of the world, and, wanted to get jacked. So that's what got me into muscle biology. And then, I sort of segued from biology undergrad to ex-phys because Rick Kreider had just taken a position at Baylor, and he was one of the first to look at the effects of creatine supplementation in humans. And I thought, wow, I didn't even know you can do research in this area. And so it just seemed like such a natural transition to do a master's under Rick. And then once I started immersing myself in the literature with creatine supplementation, with resistance training and muscle biology outcomes, I said, I just want to keep learning about this. So I didn't have any hesitation to go get a PhD. So yeah, to me, that's why muscles started to matter. It was a hobby of mine to resistance train, and I just wanted to know, how can we build muscle like this, lifting weights? But in the broader sense, Gen Pop, right? Muscle matters because we are metabolically healthy when we exercise. That's what it comes down to.
Yeah. So there's the metabolic benefits, and then some things that I've been struck by beyond just the cool aesthetic benefits of resistance training and being jacked,
Sure.
right? Cool. For example, with like fat-free mass index, if you look at the all-cause mortality literature, if someone has like a 14, so it's kilograms divided by meters height squared.
Yep.
If it's 14 versus like a 25, I believe the difference in relative risk there of mortality across a couple decades. This is apparently true from 30 years and up. It's like 70-ish%.
Wow.
So that's just the mass analysis. But then when you start looking at strength, for example, which is like often measured by grip strength,
Sure.
the upper, and this is true, I think, from 20 all the way up into like the 80s, the 20th percentile versus the 80th percentile, it's about a 50% difference in relative risk of death from any cause. So in other words, being adequately muscled and being strong, those two examples, works out to about a 50% reduction in the risk of dying from anything in a couple decades. So that's pretty cool.
That's awesome.
So, all right. Cool. So like muscle, we've built a case here, I think, that's reasonable, which is, we can take some of these basic mechanisms in culture and rodents. We can translate a lot of that from a muscle physiology standpoint to humans.
Yeah.
We think that muscle mass, muscle function is important. The differences that we see between people when they resistance train, right? We've all, Dan, I think you said earlier, you've been training for 18 years.
Yeah, about 18.
Yeah.
Since like 8th grade, 13.
Yeah, 18 years.
Yeah.
And I mean, you've put together a physique, but
thanks, man.
The 18 years, you're welcome, buddy. 18 years. How long have you been training, Doc?
Since 10th grade. Yeah. So that would be, hold on. I'm 44.
Okay.
16 and 10th grade, 28 years.
Wow.
Yeah.
Yeah. Okay. So yeah, and I've been, training about 18 years now at this point too. So considerable amount of time.
Yeah.
How much do you weigh?
220.
And you're 6'3", 6'4"?
6'2".
Okay.
Yeah.
And how much do you weigh?
Currently probably around 180, 185, somewhere in there.
So I'm about 200, 5'9". Net gain since resistance training 18 years ago, it's about 60 pounds. Not all of that is pure muscle mass. How much muscle do you think that you gained across that time?
Oh boy, good question. Ooh. I would say, thinking like in my head, DEXA data, parsing out the fat, I would speculate to guess for 30, 40 pounds.
Yeah.
Yeah, I otherwise wouldn't have had if I never lifted a weight.
Yeah.
Yeah.
And then
difficult to say. I guess if I really thought about when I got serious with bodybuilding, I was a sophomore in high school, and I had just bought Ronnie Coleman's 8-time Mr. Olympia training book.
Yeah, you did.
Yeah, and I remember bringing meals to school, and it was really taboo at that time. People are like, why are you bringing chicken and rice? I'm like, because this jack dude.
Look at this.
Yeah. And when I started that, it was maybe the end of my sophomore year of high school. I was probably 125 pounds. So I was smaller. I don't remember how tall I was. I don't know if I grew a couple more inches. So yeah, I guess 125 pounds to currently sitting somewhere between 180, 185 pounds.
That's so
But you remember when I was a master's student, your master's student at LaGrange College, I showed up maybe around 210.
Yeah.
The peak of my powerlifting days.
It was Dan with a capital D.
That's right. Big Dan. Strong. So I've lost some weight. So I'm down probably 30 pounds.
Okay.
So
yeah, that just to kind of give an idea, and that's just generally what we see. I mean, it's rare to see someone with a fat-free mass index of 25 or higher unless they're enhanced.
Yeah.
Meaning, using anabolic steroids.
I've done that before, by the way. We can talk about that later.
Yeah, well, it's a very interesting world that, is often moralized, and, of course there are scheduled things, and we don't want to get flagged on things that, when we post this, but I think there's just a lot, if it is as important as we think based on the research, and you have someone who's, let's just say, in their 60s, and there's the whole anabolic resistance thing, and females with menopause, like it becomes more challenging to build muscle.
Yes.
I do think there's some interesting clinical applications of some of those, I like to call them molecular adjuvants, that, are often demonized.
Yes.
That I think could actually, alongside resistance training intervention and a good diet, help people live longer and healthier.
100%.
Yeah.
Yeah. Sad.
Yeah. Yeah. I'm hoping with some of the changes, policy-wise, that, we might be able to investigate them at least more. Because it's a big challenge, right, to do a study on anything that's controlled.
Yeah. Absolutely.
Shallender Bassine, I know, is
Shally. You were going to post something for Shally at one point.
Man, real close.
Yeah. So those Bassine studies, I mean, just incredible. And sad, because they may have been like this black swan event that we may never see again in research because of the evolution of, the stringency at which research is regulated, which some of that is good, of course, but, we need to know what these things do.
Well, and I mean, that study, one of the first, right, I think it was late 2000s, maybe 2010, where I think there were 4 groups.
Incremental doses.
Yeah. Well, there was an incremental dose.
Oh,
then there was one that was
New England Journal, 1996. So
okay,
it was, control nothing, exercise nothing.
400 milligrams of testananthate.
And then the combination of exercise, but just taking just taking the testosterone versus the exercise group, I think it's like, oh wow, this is effective.
It was the same, right? They gained the same amount
of muscle.
And they saw similar improvements in strength.
Yeah.
Well, and the relationship between changes in muscle mass and improvements in strength, I know has been questioned in recent history, but
sure.
That is a good example, I think, of if you add more contractile proteins that when they interact, you produce force.
Yes,
it would make sense that you produce more force.
Yes. And here's the deal with Shalley's follow-up research, right, where he did the incremental dosing stuff in younger men and older men when going up to, I think, 600 mgs per week of testosterone, you saw the blood pressure, you saw what else? There was a negative side. Oh, prostate, right? The prostate hyperplasia.
Probably hematocrit, hemoglobin.
Yeah, hematocrit was high. So without that research, we're not informed, right? But what we can say is, oh wow, it looks like 200, maybe 300 has a lot of positives without the negative side effects. Yeah. So it informs.
Yeah, and then to give the listeners an idea, right? That's about, and of course it varies between individuals, but that's 2 or 3 times.
Correct. Yes, 75 mgs a week on average is what the body produced when you are peak. So yeah.
Well, I don't want to derail us too much on that, but definitely interesting. Yeah, so there's an obvious effect of testosterone, and let's say supraphysiological doses, and not all of which are going to promote negative health outcomes, it appears. But you mentioned the 600 milligram amount, which is easily 6 times the amount of what we would normally produce.
Yes.
That's why, if someone is considering that, it's a good idea to have medical supervision, monitor lab work.
Yes.
Which is part of the reason that we model Fitomics the way that we do with our esteemed Dr. David Michael Hardin and Carolee, our nurse practitioner, and we also do the Research Labs Insiders, which is a way to monitor labs. So yes, shameless plug for all of that. That said, man, with these anabolic agents, we often think of testosterone, but one of the caveats, one of the reasons we see some of those side effects, like we mentioned, the erythropoiesis, increases in red blood cell and hematocrit hemoglobin that could make the blood more viscous and stress the heart, the kidneys, liver, etc. There are others that another one is another side effect is the conversion of testosterone into estradiol by aromatase activity.
Yeah.
And that can manifest, with gynecomastia and acne, etc. So there are other things like maybe, well, I guess we don't want to get into too many specifics, but not all of those is the larger point, would promote the same side effects. They'll hit the androgen receptor and promote anabolism, but not be converted into estradiol, for example. Do you think that in the near future we will be able to investigate these sorts of compounds?
Oh boy. So I think it will be done in medical centers if they are human studies. They will be formal clinical trials, is what I think. Yeah, most of the literature that I've seen on it, because I had a huge fascination with testosterone, and being a bit of a gym rat when I was in college, I tinkered with pro hormones that used to be legal and now they're scheduled. And some of them were better than others, but there was not a lot of data aside from DHEA and some hit and run studies out there on certain pro hormones at the time, which are now scheduled. So it would be great to see them. I think they will have to be done at medical centers, hospitals, with oversight from physicians, obviously monitoring blood work. And I just hope they're done. As to why we're not seeing those studies currently being done and published, I think it's tough to say. I mean, and I haven't kept my nose in that literature too carefully, but the Shally-Bassine stuff was really, it seemed like it was a great body of literature that was done in the 90s and early 2000s. And they were building a story starting with some of the body composition changes, the strength changes. They then were getting biopsies showing enhanced satellite cell activity with testosterone, et cetera, et cetera. Some of the stuff that Steve Borst did was preclinical, but they also used patches in humans. And now it's kind of the dark ages on that stuff, which fascinates me because we know it's effective, right? So yeah, it would be great to see a resurgence in that. And the one thing I think viewership should appreciate here is that, look, right, I think I saw a stat, and I use this in my talks when I give lectures, but 10% of the current population is under the age of 65. Well, by the time we hit 2050, that's going to be 25%. I mean, we are an aging population. And that's why, frankly, I think you're seeing this push for, healthier living. I mean, you have certain influencers, and I think of Brian Johnson doing crazy biohacking, injecting rapamycin, follistatin, etc. I mean, you don't have to over-engineer it. It could be as simple as taking 200 milligrams per week of some of the stuff. And so, anyways, hopefully we get more literature there. I will say this, with the advent of advanced proteomics and deep sequencing, it would be very fascinating to replicate some of those study designs and interrogate the muscle biopsies to get a very complete picture of what's going on there, right? So, yeah.
Well, and the other fascinating thing about that is the myonuclear permanence.
Yeah,
right.
we
That
were
seems
talking
to
about that. Yeah,
exist, right? Where, I mean, I reviewed a study. I believe first author last name was Nielsen.
okay.
Nielson. We can put these studies in
the show notes. Yeah,
yeah, and they basically showed with cumulative use using linear regression, the longer that someone had used in years, the myonuclear density, so effectively the number of myonuclei per unit micron squared, was higher as long as like 4 to 10 years after terminating use.
Yeah.
And the former users, according to the body composition data, some of the muscularity data, the muscle mass, I think they used DEXA scans. They were the largest.
Sure.
And some of these folks hadn't used in like a decade.
Yeah. I think that, yeah. So the one study I am intimately familiar with was a rodent study, again, translated, but Christian Gunderson, he's in Norway, and he kept it relatively simple. He gave one group of mice a test pellet and then discontinued the pellet. The other group of mice had a placebo pellet and then exposed both of those to synergist ablation, which caused compensatory hypertrophy. And the former test pellet mice showed two things. One was this myonuclear permanence, which, for the unfamiliar viewer, right? If you think of a muscle cell, it's like taking an uncooked spaghetti noodle, pulling it out of the box. It's really long, small diameter, and it is just peppered with nuclei. It's multinucleated. And according to the myonuclear domain theory, as the cell increases in diameter, we need to add nuclei to that cell from satellite cells in order to regulate cellular homeostasis, because the nuclei contain DNA, DNA express genes, genes are at the mRNA level are translated into proteins, which, then help the cell do what it does on a functional level. So, yeah, the fact that you have myonuclear permanence, even when the muscle size is reduced following testosterone use, would then imply that we have muscle memory at play, so that when you again, expose it to an anabolic stimulus, but this time instead of drugs, it would be mechanical overload or resistance training. That cell with more myonuclei can now mobilize all of the myonuclei to express certain mRNAs needed for hypertrophy, which then get translated into proteins, and they can accrete in the form of myofibrils or certain metabolic proteins needed to help the cell function under the stressor of exercise. You know, I don't think that's far-fetched. I think
Right.
it actually is true, so that's a slippery slope topic, because then you say, well, heck, if we talk about athletes that formerly used and they were caught using, should they be banned for life? Don't want to get into that. But it certainly is an interesting topic.
Right.
Right. So, but just when we talk about personal health, I use pro hormones. I tell my students all the time, I used to dabble with an anthate, et cetera, et cetera. And I feel like if I ever take time off from the gym, because of work and kids and all that stuff, when I re-engage in the gym, it is not hard for me to get to where I was in terms of muscularity and strength. And of one anecdote, I know, there's limitations in that, but there could be something there. So very fascinating. And I would love, I'm sure the human study you reviewed was probably survey based, wasn't terribly mechanistic aside from getting the biopsies, but didn't have a predefined dosing regimen followed by a well-defined, we're going to take this much time off. I mean, that's essentially how we're going to answer that question, right? And then it'll inform us moving forward. From an aging perspective, right, I think that's very fascinating because, let's say you reach peak testosterone production in your mid to late 20s, and then you're on the downslope of andropause, and perhaps you take replacement doses for, we'll call it arbitrarily 5 years, but then maybe you discontinue that. Can that permanence last 10 years plus and be a continued aid for you when you resistance train? Fascinating, right?
Yeah.
But again, we don't know.
Gotta do the research.
Yeah.
But
We need to do the research on that.
It appears promising. Like this, perhaps even like a phasic or cyclical approach.
Sure, 100%.
Deserves investigation.
Yeah, I agree.
Yeah, but there's enough evidence that there's promise there, it seems.
Yeah, I would agree with that.
Well, and then Doc.
Yeah.
We've of course got to talk about.
I know it's coming.
Yep. Right. So the cell
Sarcoplasmic hypertrophy.
Here it comes.
Here it is.
So the cell gets bigger.
Yes. So when you resistance train, think about the muscle cell looking at it in cross section. Because if I turn it this way, it's that long spaghetti noodle. And this gets into Danny Plotkin and some of his interests. He's also interested in lengthening contractions causing longitudinal hypertrophy.
Sure.
Very hard to assess, by the way, in a human model.
Right.
But let's turn it in cross section for a moment. Very well documented when you lift weights and you consume the proper amount of calories, you're going to have this radial hypertrophy of the muscle fiber. Now, get a microscope and look inside that cell in cross section, right? What do we see? We see mainly myofibrils. 80 to 85% of that cell contains myofibrils, which contain the contractile proteins that help the muscle cell function as, this cell that can contract. And through tendon linkage to the bone, this is how we move our limbs, etc. All right. What happens when you resistance train? Well, again, very well established. We have this radial hypertrophy of the cell, that we have myonuclear accretion that I mentioned from satellite cells, basically fusing to that pre-existing fiber. But what happens to the myofibrils and the non-myofibrillar space is certainly a topic of contention, and I don't have a fine pulse on it, but one of my students that y'all both know, Danny Plotkin, he's seen a lot of chatter recently on social media, some of which comes back to
and everyone knows
Haun et al. Papers
talking about, right, and has actually developed the assay to isolate myofibrillar proteins. So that was
sure.
you, actually.
Yeah.
I
So
think you, right?
yeah, so
that didn't exist before.
We had a method that was called the MIST method, and our motivation for doing that was actually spurred by some of your papers.
Goldspink, right?
And Jeff Goldspink had done some of this stuff. Wow, with myofibrils, he was using a few things, one of which was crude light microscopy, which I would not recommend because the resolution of looking at myofibrils is insanely bad. And me and Troy Hornberger have talked about that. And I'll bring up Troy in a moment because he had a critical breakthrough recently on myofibril imaging not involving TEM.
Right.
Okay, so how can you measure this? I think is what we're sort of getting at here, right? Biopsies, pre-post resistance training. You can use one of two imaging techniques. One is called TEM, very laborious. Some critics would indicate that you have artifact from tissue fixation, dehydration, embedding with resin, et cetera, et cetera. And oh, by the way, when you get down to that, we'll call it 10,000x magnification, you're looking within one cell, right? And we know that the VL, which is what we biopsy, can have upwards of 500,000 plus muscle fibers. So the limitation is obviously sampling there with TEM. You can use this FemID, which is fluorescence imaging that Troy recently pioneered from his laboratory, and we have a paper on, we can link in the show notes, where you stain the tissue section from the biopsy and cross-section for SERCA1, SERCA2. So SERCA1 is a protein, the sarcoplasmic reticulum protein, that's found exclusively in type 2 fibers. SERCA2 is found in type 1 fibers. And through these outlines, the implication is that's going to size and number the myofibrils. And we engaged with Troy. He was super fascinated with the Haun et al. Review, which you should link in the show notes. When we, we were pontificating, what is hypertrophy? Because boy, the methods to assess it don't align. And there was what was his name? I think Eric Gordon was a researcher in the 60s saying, well, heck, tissue weights after exercising rodents doesn't align with the percentage increases in muscle fiber CSA. Troy, and then Goldspink and some of his work indicating that we have an increase in the size of myofibrils with radial myofiber growth. So along the same time that you wrote that review with myself, Brad Schoenfeld, et cetera, Troy was on the hunt, with the same topic. And so we finally said, hey, Troy, look, man, we're going to do a resistance training study. We know you're doing this in mice with the weight pulling model. Why don't we send you some biopsies to do your FemID? So we did. Come to find out, what he found is when you take somebody that is untrained, right? And in our study, it was 7 weeks of intensive resistance training. I think the ultrasound indicated that we had around 20% VL cross-sectional area increases, so robust. He found that the fiber CSA increases were due to an increase in the number of myofibrils, but not the size. And so that is the best model we have to date, because Goldspink said, well, no, it's the increase in the size of myofibrils, not number. Limitation was he used light microscopy, alpha resolution, and then the other stuff came from J.D. McDougall at McMaster in the 1980s.
And that was TEM based.
And that was TEM based, where you're sampling probably 1/1000th of the cross-sectional area of one myofiber.
But he did at least appear to show that.
Yeah. Okay.
Yeah.
So the caveat here is if you start lifting weights and you hypertrophy, what's going on in your muscle cells is we think that you have this increase in the number of myofibrils, but you don't see what we would term as sarcoplasmic hypertrophy, which would be this disproportionate increase in the cell size relative to the increase in the myofibrillar space, right? And that is to say, if I lift weights and my muscles get bigger because of sarcoplasmic hypertrophy, it's because of the fluid and the non-myofibrillar space inside of those cells increasing disproportionately to the myofibrils.
Even things like the circa.
Yeah.
Right,
Because
the
the circa,
sarcoplasmic reticulum,
yeah,
rather, and
yeah.
mitochondria, ribosomes,
Yes. So why did our lab start this research? It was based on your dissertation. And to set up the plot for that, your idea, which was brilliant, and it was based on more macro related questions at the time, was, can we take a bunch of trained, resistance trained gym rat type guys and split them into 3 groups? One group is going to take a placebo. One group's going to take a standard scoop of protein per day. One group on a weekly basis is going to scale the amount of whey protein, which is to say, during week 1, I would take 25 grams of whey protein per day. During week 2, I would take 50. During week 3, I would take 75.
Mm-hmm.
And by the end of the study, I was taking 150 grams per day of whey protein, right? Now, what also happened was we scaled volume. And that was an intriguing question because it's, we all know that, okay, well, if we supplement the diet with protein, right, 1 or 2 scoops per day is great. And that should help support our training adaptations. But what happens if we scale volume? Do we need more protein to continue to see growth? Come to find out, I think you took per major muscle group, we were having them perform 36 sets per week, which is astounding.
Per exercise.
Yeah, per exercise.
We had a little bit of variation on the midweek session, but around 35 sets a week for 4 exercises.
Yeah, so just to let people know, I mean, really take pause. That would mean you would come in Monday, Wednesday, Friday, and grab the barbell, get under the barbell on bench press Monday, Wednesday, Friday, and do 12 sets of 10. Insane, right? And as a matter of fact, I mean, this is sort of tangential to what we're talking about at the molecular level, but I tend to settle on 20 sets per week. Certainly is probably all you should be doing, and perhaps you'll have different insight on that.
Should.
But and I would say that's probably the max, right? I mean, at the time, before we got into your study, I think Schoenfeld's metadata would indicate that like at least 10 sets per week. It was something weird to where it's like we saw continued growth from his metadata all the way up to 10 sets per week, but there wasn't anything well defined past that. So I tend to tell people, based on what Cody did, and I think DeSouza at Tampa recently did a study. It looks like 20 sets per week is the sweet spot. Anything more than that, not necessarily inverted U. You're just not, there's a point of diminished returns. Now, what does diminished returns look like? Well, in your study, in doing the BIS, which looks at intracellular, extracellular water, and doing that in conjunction with DEXA, what we see is up to 20 sets per week, this sort of linear increase in lean tissue mass gains that wasn't disproportionately affected by extracellular water, right? When you go 24, 30, 36 sets per week, yes, the DEXA will say you are continuing to put on lean tissue mass, but, right, a lot of that could be driven by extracellular water. So that has started what seems to be a very hot topic on social media, that, training with too much volume or even training in general and the hypertrophy they're in is largely edema. And then we go to the Haun et al. Sarcoplasmic hypertrophy study, and that's more evidence of edema. I think that's a gross misrepresentation of the data in the sense that we took trained individuals that were probably at a ceiling in terms of hypertrophy. We then had them do this very unaccustomed high volume based training. And yes, there could have been some swelling, edema, sarcoplasmic hypertrophy, but by and large, you're not going to see that in Gen Pop. When do you see sarcoplasmic hypertrophy? I think Haun et al. Would be a good study, a candidate study for that. I'm intrigued with the notion of creatine loading and supplementation, because we know if we all sit here for 7 days and crank on 30 grams per day of creatine pre-post DEXA, our lean mass is going to be up 5 pounds, right? And so I'm not convinced that that's all myofibrillar hypertrophy, meaning that 5 pounds of muscle is due to myofibrils increasing in number. So I think that's fluid driven, and that, if you were taking biopsies and looking at the ultrastructure, you're going to see some sarcoplasmic hypertrophy. 600 mgs per week of testananthate. So probably some sarcoplasmic hypertrophy. And as a matter of fact, that JD McDougall 82-83 study, we talked about this offline, but I believe some of those folks were certainly power lifters, but I think some were steroid users as well. And you saw that phenomenon. So, it's still a question I'll get routinely. I know you talked to Mike Isertel about this, and we talk about it, and me and Stu will talk about it. Stu Phillips, can sarcoplasmic hypertrophy occur? Yeah, I think it does. I think if you hit your natural max potential, which in and of itself is another conversation, and then you do something to try to bust through that, meaning like, okay, I'm going to take testosterone, or I'm going to jack my weekly volume per muscle group up to 36 sets per week. I think it can happen, right? We just need to do a little bit more literature there. And so working with Troy Hornberger, his FemID technique is certainly a robust one. And I think eventually we'll collaborate on a project like that. But I'm not one to say that sarcoplasmic hypertrophy doesn't occur. Aside from your study, Chris Van did a follow-up, which you were a collaborator on. Within subject design, trained individuals, one leg was exposed to high volume training, one leg was a high load training regimen, pre and then post intervention, MRI, biopsy. We did not do histology, but we instead did the 6-week intervention integrated myofibrillar and sarcoplasmic protein synthetic responses. And what we saw was the high volume leg hypertrophied according to the MRI. And in addition to that, the high volume leg had more sarcoplasmic protein synthesis over that entire 6-week period compared to the high load leg, again, supporting the model. So, yeah, I mean, we need to further investigate it. Now, practically, does it matter?
You're
right. So, I think yes, in the sense that maybe it is an indicator of your natural potential for your muscle cells to accrete muscle proteins, right? So I think at a certain point, those cells reach a size limit, and if there's any additional growth within a small window of time, it's going to be more sarcoplasmic hypertrophy. Does that make sense?
I think it makes sense. And I mean, shout out to you for you snuck unicorn into that title for Frontiers on this topic.
Yeah.
Is it a unicorn? Maybe. But those ideas, I mean, they make sense to me. That that's the priming. There's the spatial priming,
there's
the bioenergetic priming, and then there's like the biomaterial or the actual ribosomal, content to prime the cell to eventually backfill, if you will, with myofibrils.
Yeah.
And I mean, from an engineering standpoint, like we're going to build a structure. We prepare the space. There's grade work involved, right?
Yeah. No, remodel the cell so that it can
Right.
then be filled with what it needs to be filled with. That makes perfect sense.
Makes sense. And I mean, even if, for example, in the steroid scenario, some amount of that is sarcoplasmic, well, if they continue to use it and train, they appear to continue to gain myofibrils.
Yeah.
So you know what I mean? I think there's a temporal aspect to this whole thing where it's not dichotomous. It's not either sarcoplasmic or myofibrillar. It's probably more of a continuum
100%.
or a spectrum, depending on a variety of factors, the training stimulus, right? Any molecular adjuvants, supplements, diet, etc.
Yeah. And along those lines, what I also, and this is a highly contentious, issue, is the splitting of the muscle cells themselves.
Oh yeah, I mean, then you get into like, is it hyperplasia actually a thing?
Right.
You think it's splitting?
I think it's splitting. Now, you and I, when you were a second year in the lab, you came to me asking that question. I said, no way do you see splitting or any hyperplasia. But after reading further literature and then performing IHC, so looking at tissue sections under the microscope, you can see features where you have a large cell and part of that membrane is invaginated. It almost looks like the cell is actively splitting. And Lars Ericsson, they published data again from steroid users, self-reported, showing that there were more cell splitting events from those muscle biopsies versus people that were non-users. And, so the prevalence is not that high, but it's a continuous process. If there is the double of the amount of splitting of muscle cells in steroid users versus non-users over time, that will lead to appreciably more muscle cells, right? So I don't think we can necessarily dismiss that mechanism as well. And muscle is a fascinating tissue.
Yeah.
In particular, when you study it in the context of a stressor like exercise,
right?
Because of how adaptable it is, right?
Yeah. That's cool stuff. Did you have any follow up to that?
All right. So, circling back to Haun et al. With sarcoplasmic hypertrophy, what led you to the conclusion that this was likely sarcoplasmic hypertrophy that was occurring, as opposed to maybe more of the classical model of being myofibrillar?
You take a stab?
Well, I was following your lead on those assays, of which you went on to, I think is it in Journal Biology, where you actually
biological
published
methods
the
where
isolation.
Wanted to make sure the isolation was robust, which it
Basically,
was.
had to develop a custom assay to help answer the question.
Right. So I remember Cody. We finished the project, long project. That fun story was one of the only projects where I went to help train participants because I was so concerned with the high volumes, being 36 sets per exercise per week, thinking that was going to injure somebody. And I wanted to be a witness when I had to go to the IRB and tell them why people were getting injured. And no one got injured.
Yeah.
And oh, by the way, the hardest week of training was week 1, where people were yakking in the hallway and outside the building. And,
Shout out to those guys, troopers.
Yeah. And what's crazy, I think week 1 we started at, help me, Cody. The midpoint was 20 sets per week per exercise.
I think it was 10 the first week.
10, okay. Because it was 3 sets for Monday, 4 sets for Wednesday, 3 for Friday, and people were yakking because they had to do that for squat and bench press and lat pulls and deadlifts. Brutal. But what was crazy is by the time they got to week 4, 5, 6, They were just honed in. They were conditioned. They got through it.
Locked
in. So anyways, we have pre-intervention biopsies, mid-intervention biopsies, post-intervention biopsies. It was a 6-week training block. Cody and Dr. Chris Van were spending a lot of time in the laboratory. Part of the plan was to do histology. I think we were going to do some satellite cell stuff because, again, the original intent was to look at, okay, in one of these groups, we have this incremental increase in whey protein per week. One group was standard whey protein. One group was placebo. Come to find out, didn't matter what they were taking. And so we just said, okay, now with all 30 some odd participants, let's just look at significant main effects of time because this is an interesting volume load model, right? So Cody and Van, they took the pre, they took the mid, they took the post biopsy, and there was actually a fourth biopsy, which was after a 1-week deload. We won't get there. Maybe another episode on a podcast, but pre, mid, post, and did histology. Think we were going to look at fiber type specific hypertrophy, myonuclear number. With the non-histology tissue, I don't think it was relatively well defined at the time what we were hunting for. Perhaps I think we were going to look at some proteolytic markers. We didn't do tracers in that study. We were going to look at certain markers like ribosome biogenesis and ribosome content, et cetera, et cetera. And this was Cody's idea. He was interested in looking at just the myosin heavy chain pooled, isoform content. And I don't even know why, but it turned out to be a really good idea. So he comes with that data, and he comes with just the total sarcoplasmic protein content data, and says, Doc, sarcoplasmic protein content is going up, and it looks like that the myosin heavy chain pooled isoform content is going down from pre to mid and mid to post. And I said, I don't know. If anything, based on how that assay works, we should see no change in those values because they're all normalized on a relative basis. It's almost like you're getting the same amount of muscle pre, mid, post, and so your stoichiometry works out to where you should see no change in the value, and that would be supporting conventional hypertrophy. So I think that's when we started digging, and we found J.D. McDougall, 1983. Oh, wait, it looks like that you can have this dilution of myofibrils in these power lifters. There was another study by Meyer et al., where it was a cross-sectional comparison of off the street controls that don't exercise, recreational weightlifters, bodybuilders, and when they isolated single fibers, it looked like that the specific force, which would be the force generated normalized to the cross-sectional area of the fiber, was lowest in bodybuilders, which would imply that there's, on a relative basis, less of the space occupied by myofibrils, and that's where we started. And so that led to us publishing that sarcoplasmic hypertrophy paper by Haun et al., I think in PLOS One, where we were interested in just looking at people that showed this increase in fiber CSA to the protocol, which is like half of the folks. Now, the one critique that we got was, well, why didn't you look at folks that didn't show this increase in fiber CSA? There really is no good reason why we didn't, except to say, well, okay, if this extreme hypertrophy caused fiber CSA increases, we just wanted to see within the people that showed that phenotypic change, what was going on. Was there sarcoplasmic hypertrophy, et cetera? And all signs indicated that, yeah, we saw features of what we would consider to be sarcoplasmic hypertrophy. Looking back, it would have been great to implement Troy's model, his FemID, because I do think that is a more robust imaging technique than what we use, which is the phalloidin stain. Because this is technicalities here, but when you're looking at this tissue under a microscope, phalloidin staining is a technique where this compound, that's conjugated to a fluorescent dye, it binds with high specificity to actin. And so you consider that as, okay, we're looking at myofibrils that fluoresce because that's, myofibrils have a ton of actin. There's some structural actin there at the cell membrane. But anyways, you can't get the number of myofibrils or the size of myofibrils, but you can get the relative content of myofibrils with those sections using fluorescent staining. Implementing something like what Troy does, we would have gotten the number of myofibrils per muscle fiber, the size of them, right? And then you can look at the space between them. So, do I want to replicate Haun at all? I don't, I guess if we have to, if we have like somebody, an influencer out there that really wants to answer the question, we do it again. But yeah, I think as we evolve, and this is sort of a tale of just research in general, as we evolve, I mean, it's been almost 10 years now since that study, we have better techniques. And so, we can use these molecular tools in the toolbox to better get at that question. Yeah.
Well said.
Thanks. Did I answer your question?
Yeah.
Cool. Okay.
Yeah.
Well, with this mode of hypertrophy, we'll call it, so like there's potentially this, let's say, continuum of more or less sarcoplasmic versus myofibrillar.
Sure.
If we zoom out,
yeah,
we also see considerable variability in how much muscle is gained in response to resistance training.
Yeah.
Right. And this is also a controversial topic I have found, as far as how you analyze this data.
Yes.
But I think just like the observation in the practical setting is useful here, where it appears some people, despite high effort and doing a lot of, I would say, the right things, eating adequate protein, using a reasonable training program, just tend to have a harder time gaining muscle than others. You've written extensively, thought a lot about this. Just to run the list real quick, because I'm curious about where your head's at now, if you've learned anything new, if you're looking to interrogate new areas.
Well, yes. And by the way, I'm a co-chair for a symposium we're going to do in Finland this year, November. Shout out. Juha Tienen and I. Yeah. So he's the chair. I'm the co-chair. We're going to have Marcus Baumann, Stu Phillips, Troy Hornberger, Gustavo Nader.
What a crew.
Adam Sharples.
Jeez.
So yeah, if y'all can make it to Finland.
Yeah, that'll be a heck of a conference.
Yeah. But yeah, it's certainly, and Atienen, so he's at University of Jyväskylä in Finland. He is running quite a large response heterogeneity trial that's going to take 3 years.
Nice.
And so our lab, Marcus's lab, we're going to help him analyze bits and pieces of that tissue to get at some of the currently known mechanisms which seem to drive response heterogeneity, one of which is ribosome biogenesis. It seems like if you can pack muscle on in the gym, when you lift weights, there are signals in your muscle cells that cause those cells to generate more ribosomes, which then facilitate protein synthesis, right? And the other mechanism, which has popped up time and time again, we've shown it in females. Stu Phillips has shown it. There's details there, but Stu's shown it. Marcus Baumann, most famously, I think, demonstrated this, was that people who activate and mobilize satellite cells for myonuclear accretion tend to be your high hypertrophic responders. So again, two things to recap: ribosome biogenesis and satellite cell mediated myonuclear accretion seem to be, at the molecular level, what drive people to gain muscle in the gym, right? Yeah.
Well, and I know you've taken a look at this since we wrote the review article in Frontiers, but copy number repeats. Well, let me back up.
Genetics.
So there's ribosomes, the molecular machines that translate messenger RNA into proteins.
Correct.
That is protein synthesis.
Correct.
The more of those molecular machines that we have in the cell, our translational capacity, you could say, is greater.
Yes.
So that makes sense. Where do the ribosomes come from?
Yeah, good question.
And so there's copy number, potentially repeats, and if you maybe have more of those, what do you think about that?
Yeah. So think sort of use your imagination. We're looking at the muscle cell in cross section. Again, mainly myofibrils. Between myofibrils, you have very small macromolecules, ribosomes, that translate messenger RNA into protein. You can also have some of these ribosomes localized to the myofibrils. So you can have, free floating, or I guess ribosomes suspended in the sarcoplasm, but you can also have myofibrillar ribosomes. And so the current thought, this is actually out of Eric Wang's lab, Florida, when your myonuclei export messenger RNA, they don't just sort of freely float around and randomly bump into ribosomes. There is a trafficking system for that. So some cytoskeletal proteins will, using molecular motors, will literally take these messenger RNAs either to the non-myofibrillar ribosomes or the myofibrillar ribosomes. And then from there, you can have the translation of proteins, and you can have the deposition of the proteins where they belong in the cell. Where do they come from? Where do ribosomes come from, right? Just like any other protein in our cell, genetic code. So these myonuclei, right? They're transcripts, ribosomal RNA that are produced from transcription. Okay. If you lift weights, you will stimulate that transcriptional process. And from there, within the nucleolus of the nucleus, you have the assembly of ribosomes, and then they're exported and they're trafficked either to the sarcoplasm or to the myofibrils. Okay, so this is interesting because the gene that's responsible for ribosomal RNA production, and again, ribosomal RNA eventually gets integrated into ribosomes, is called the rDNA gene. All of us sitting in this room, we have it's not like alpha-actinin 3, myostatin, IGF-1, where every nucleus has 2 copies of that gene. You can have dozens, if not over 100 copies of the rDNA gene. That emphasizes the importance of ribosomes for cell homeostasis, right? The fact that you have multiple copies spanning different chromosomes. The cell needs to make ribosomes to survive. A group out of, I forget where, but I'll go here. So Kentucky, they were the first really looking at this in the context of skeletal muscle hypertrophy, and their preliminary work in humans, it was from like 7 or so humans, indicated that individuals with more rDNA copy number showed enhanced ribosome biogenesis in response to one bout of resistance training. So we found that quite interesting because we are from the school of thought that, well, heck, if you can have enhanced ribosome biogenesis due to multiple bouts of resistance training, you should be a high hypertrophic responder. So Josh Godwin in my lab, he's now postdocing with Nader, looking at ribosomes. But we took a cohort of 80 individuals that had done 10 weeks of resistance training with us. We had pre and we had post biopsies. We isolated DNA to get relative rDNA copy numbers. So how many copies of this rDNA gene do you have? And then from there, we wanted to see if there were associations with hypertrophic outcomes. And we saw none. So what does that mean? Well, we think if you're an individual that per myonucleus has, we'll just call it arbitrarily 20 copies of the rDNA gene, right? And I'm an individual that has 40. It's not necessarily the number of rDNA copies that we have. It's how we mobilize them to be transcriptionally active. That's more important, right? That would dictate the hypertrophic response. So that's where I am there in terms of if we talk about genetics and their influence on hypertrophy and being a high or a low hypertrophic responder, I'm not convinced yet that RDNA copy number is important. And as a matter of fact, in that same paper, Josh showed that you can chemically actually knock out some of these DNA copy numbers, and then we can stimulate muscle cells with like IGF-1. And if we knock out some RDNA copy numbers in some cells, and we don't knock it out in other cells, and then we treat both of these cells with IGF-1, they hypertrophy the same. So that sort of mechanistically proved the point that we saw at the correlational level in humans. All right, so genetically, I'm sort of, with a pencil crossing that off my list. Are there other genes? Are there other polymorphisms?
Or perhaps epigenetics.
And we'll get into epigenetics, but are there other polymorphisms that may explain the hypertrophic response? So Eric Hoffman, they ran a large, multi-year, probably a decade clinical trial. It's called the FAMOUS study. And at the time, they didn't use microarrays like we did, because I'll get to that study in a second. But they used just single nucleotide polymorphism approaches using qPCR. And they looked at 500 different types of polymorphisms. And for the unfamiliar viewer, if you have a genetic polymorphism, if you're a certain genotype for, we'll call it alpha-actinin 3, that's a famous one. 23andMe says this is the athlete gene, which is grossly oversimplified, by the way. But you can be one of two genotypes. You can be the R genotype or the X genotype. All right. Alpha-actinin 3 gene, if you are XX, that means that you have two versions of that gene, one from mom, one from dad, where you produce a truncated version of the protein that becomes degraded. So you don't have any alpha-actinin 3 protein in your muscle cells or any other cell in the body, right? There have been associations. Steve Roth did this years ago with power lifters at the competitive level. None of them are the XX genotype. They have at least one copy of what we would consider the functional alpha-actinin 3 gene, right? And so that led to a ton of excitement saying, we can use genetics to maybe predict an athletic phenotype. It seems to hold up. It's held up reasonably well in the literature that, it seems like a lot of the competitive powerlifters, weightlifters, et cetera, have at least one copy of the functional gene. But when you look at Gen Pop, the responsiveness to resistance training, et cetera, from my read of the literature, it's not that impressive. When you look at hypertrophic potential, whether you're XX, RR, or RX, doesn't seem to predict hypertrophy. So I've crossed that off my list, right?
But that's one.
That's one gene out of the 20 plus thousand we have in the genome. So we've crossed two off of our list.
Right.
Our DNA copy number is a source of genetic variation.
Perhaps we use, here it comes, omics.
Yes.
High throughput.
High
Right?
throughput.
We get all the genes, all the proteins, all the transcripts, all the metabolites.
Yes.
Fit omics.
Yes. Yeah, exactly.
There it is.
There it is. So we used omics. We collaborated with Stu Phillips, and we said, Stu, send us muscle that you have and DEXA data that you have from any training study that's 10 to 12 weeks. What we're going to do is we're going to isolate DNA, and then we're going to look at the pre-post intervention fiber CSA changes, as well as the pre-post intervention DEXA changes from your cohort, our cohort. We're going to pull it together. We're going to send the DNA from all these people off to do what you call a DNA, SNP array.
This is a GWAS.
This is kind of a GWAS approach.
This is Rupel. Wasn't he the first author on that?
This is Chris Van.
Oh, Chris.
Yeah. So
Chris, the ring finger.
Right? This is Glee 3.
Okay.
Glee 3. Yeah. So long story short, we did this DNA microarray, 120 some odd subjects, hoping that we would find a candidate gene which predicted the hypertrophic response, either with fiber CSA or DEXA lean tissue, and come to find out we didn't.
Right.
Now, there was one, we adjusted our statistical approach to have a less stringent p-value, and there was one annotated SNP that was associated with a gene that's called GLE3. GLE3 is a transcription, the gene encodes for a protein that acts as a transcription factor. It seems to be involved with satellite cell activity. So we said there may be a little bit of promise there, but again, it could be a spurious finding based on the number of SNPs that we looked at. So where I'm at with genetics is we have to use sequencing.
Yeah.
And we have to do modeling because it's probably a combination of different polymorphisms or different things such as microsatellite repeats. Like the androgen receptor is a gene that can have what are called these microsatellite repeats, where there's just triplet repeats in the code. And if you have one allele, you produce an androgen receptor that has this altered protein structure versus the other allele. And so that probably affects the function of the androgen receptor. So it has more or less binding affinity to DHT or testosterone.
Makes sense.
And that could affect the hypertrophic response. Point being there is we need to use advanced sequencing and get more than 300,000 random spots in the genome.
Right.
And from there, computational modeling, we're doing a little bit of this with Marcus Baumann right now, completely unrelated. But we did a lot of different omics with disuse, and we want to predict, okay, with muscle loss after being 2 weeks in a cast or a boot, right? What are the molecular signatures that predict that response? Well, this lasso regression machine learning approach is allowing us to take into consideration phenotype. So, coming into the study, what was the lean body mass index? What was the strength? What was the size of the VL? What were the size of the muscle fibers? What were baseline expression patterns of mRNAs, all the proteins,
Nice.
et cetera? And then in addition to that, plugging that into the model along with change scores, we're going to attempt to say, okay, this seems to be the most important feature here in terms of predictive, value.
That's exciting.
Yeah. So, and that shout out to Max Michel. He really has spearheaded this, and this is going
Right
to
on.
be the crux of his dissertation.
Nice.
But sort of springboarding back to genetic factors that predict hypertrophy, right? Can we leverage the data that we have on hand for some sort of gene test that we can do in Fitomics? We're not there yet. That's my
Right.
personal opinion. I think we're going to get there, but we need to adopt more sophisticated sequencing approaches versus DNA arrays. We need to use machine learning like this, what Max is doing with Marcus. And then perhaps we can have certain algorithms based on age, based on sex, etc., that say, okay, if you have these polymorphisms, these dozen polymorphisms, then perhaps you won't gain as much muscle, and so we need to adopt an alternative response. And what I will say is this, right? A hypertrophic non-responder, Clayton Labardi, we've collaborated with that Brazilian lab. He does great work. Love Clayton. Shout out if you're watching Clayton. But they showed recently with Marcus's group that a non-responder to relatively low volume in one leg will be a responder if you 3 or 4x the volume in the other leg. And so I wouldn't be deterred because I think folks tend to say, and they say this barely working out. I used to personal train. I know all of us personal trained at one point, but you would have the Debbie Downers that would say, I shouldn't even lift weights because I'm what they call non-responders, based
A
on
hard
these.
gainer.
Yeah, I'm a hard gainer. I don't think you should, it's like, no, we need to cater the program towards
Right.
what you're going to respond to, if that makes
And
sense.
monitor if it's actually working.
Yeah, exactly.
And I mean, a lot of times, right, Dan, and like in our experience, it's if you evaluate that's like our process is like evaluate, do the assessments, take a look at the nutrition.
Sure.
And more times than not, it's like there's a lot of areas of improvement
Yeah.
to elicit a response.
Yeah.
The dosing of volume to the area that you would like to see change is one of the more common things that we would address on the front end.
Sure.
And it's like, for example, it's like, I really want to see growth in my pecs or my arms. And you look at the programming in recent history, if someone even has logs and is systematically monitoring it, it's usually a case of under or overdosing.
Yeah.
And then, don't forget the nutrition side of things.
Yeah.
I mean, one of the most striking figures I saw, like in the notes, was from Morton and Stu's lab on the meta-analysis with the 50 studies and on protein intake and the change in fat-free mass. And it's like, if you are consuming less than like 0.6 grams per kilo of body mass per day and lifting 3 times a week for 3 months, that data suggests you will gain zero fat-free mass. So just by going from there to approximately 1.6 grams per kilo, based on that breakpoint regression, perhaps it's higher in some individuals, but like that could take you from 0 to 2 kilos within that time frame. That's 4 or 5 pounds.
Yeah, and Bill
just
Campbell,
by consuming adequate protein.
That's right. And Campbell, I was going to say Campbell and Lane Norton, they collaborated on that physique female bodybuilder. I think they were doing show prep, and one group was assigned to, I think, 1 gram per kg per day, the other 2.3 grams per kg per day, and that latter group, 2.3, I think, gained 3 or 4 pounds on average more muscle.
And Jose Antonio, right?
Oh man, yeah. He'd take them up to 4+ grams per kg per day of protein. And so you're going to see the optimized hypertrophy response at those levels, as well as, and we've seen this, the loss of body fat.
Well, and right? Like in the dissertation, I know we didn't see a big change in the graded protein group, but they did lose more fat.
They did. Yeah. And Stu Phillips has shown that. He calls that the Holy Grail study. What he called, I think, the moderate protein group, which was like 1.6, and then there was like close to 3, in the high protein group, 3 grams per kg per day, hitting them with 4 days per week of resistance training, like 4 or 5 days per week of HIIT training. And I think they did this for maybe 6 weeks.
Yeah.
And that latter group, again, the very high protein group, lost somewhere on average, I want to say it was well over 10 pounds of fat mass while gaining more muscle mass.
Yeah.
So, it's hard. It's hard to do. I've really thought about those data as of late. Then you compare, you sort of juxtapose those data saying, okay, definitely high protein for optimization. But oh, by the way, anabolic resistance with aging. You need more protein because the muscle cells mechanistically cannot sense these amino acids to stimulate muscle protein synthesis. So I've gotten to the point, man, where I'm buying ready to drinks. They're expensive, but it's a good investment in health, right? Because we talked about this, Daniel, like it's hard to sit there and prep chicken breasts or, pork loin or whatever on a daily basis. Like I can just this way, 4 or 5 of those per day, that's 150 grams, eat some eggs in the morning, have a protein at night. And I mean, I'm well over 200 grams for the day. I weigh, 100 kilograms on the dot. So that's really the only way I can do it. But,
and Israel, they just released that Genius shot. Those are they're pretty good. That's like 2 ounces, 23 grams of protein, no sugar.
Oh yeah.
It's easier than ever, I would say.
Okay.
To hit the
exactly. But yeah, you got to be deliberate, right? I mean, it's got to be consistent. So
yeah. All right. That was good. Well, where do we want to take this from here? We have covered a variety of very interesting topics. I think I would be curious to know, because like the part of our mission is to help people take this heavy, mechanistic information, omics stuff, like all this advanced literature, and practically apply it to help them be healthier, live a longer life, perform better, look better, all those things. Say this on a regular basis. When feasible, like we want to test, we don't want to guess. A lot of this stuff can be measured. We can be analytical about it, and then we can take that data, we can action it. What do you think about that? Like, how could we're not there yet on genetics.
Yeah.
I will say, like I did sequencing.com, it's wild that, what was $1 billion 25 years ago?
Yeah.
You can actually get all of your genes sequenced for less than like $500.
It's crazy.
But that's just the source code. So expression, right?
Yeah.
The epigenetic patterns.
Yeah.
That's a whole layer. Like it's wild to me that you guys did that, GWAS, and found maybe there's one here. So there's other things going on. There's other assessments. I mean, we do a lot of labs.
Yeah.
I think that's really informative.
Sure.
Beyond just looking at someone's diet, looking at their training program, their body comp, their metabolism, some of our core assessments.
Yeah.
Labs are informative, blood work, just
Yeah.
blood biomarker testing and optimizing things there. Do you have any insights, ways that we could, because it would be cool if we could get a little sample,
Yep.
saliva, maybe a little blood, maybe some hair, stool, those sorts of things, and have this physiological profile of someone that is accurately measured, and then take that and prescribe exercise, maybe leverage, therapies, molecular therapies, and customize a nutrition plan for them.
Yeah.
That's really what we're working towards.
Yeah.
What do you think, man? I mean,
I think it's a great approach. And what I was going to say, with a lot of our data and a lot of studies across the board, right? I mean, y'all's enterprise has people that are bought in well, and they will do, even though it's not live-in studies, they're probably going to be compliant. More so than I would say we see at university studies. I mean, let's face it, college age kids, there are a lot of degrees of freedom outside the laboratory that can influence the adaptive response when you look at the food logs, or when, and Stu actually linked alcohol consumption with a decrease in muscle protein synthesis following a bout of exercise. And we won't go down that rabbit hole, but it's even led me to, I like wine, but I have noticed when I am routinely consuming wine, I just don't feel good in the gym. I feel like I'm not adapting. I feel like I'm more sore. So, all of that is to say that we do our best in the academic setting when we run these studies, but these are not, when we publish the paper, you are seeing the best presentation of the data, and we try to list the limitations. Whereas here at Fitomics or other places that do what you all do, I think people come to you because they seek help, guidance. They're going to buy in. And so y'all's data is probably very informative, right? In terms of tracking what matters, what doesn't matter, etc. And so I would be excited to collaborate if y'all have, we can develop a retrospective IRB based on y'all's years of longitudinal tracking. That would be incredible data to say, yeah, it looks like these biomarkers matter the most, right?
Mm-hmm.
And I think that would be even more valuable than some of the data that's published from these training studies that are done at academic institutions. So, but right now, what matters? I would say genetics matter, even though I've been a Debbie Downer on our DNA copy number and our DNA microarray data. And that's because the heritability studies do indicate, without using molecular techniques, these are like twin studies or family-based studies. Yeah, Bouchard did it famously with the heritage study. But it seems to indicate that whether you're talking about increases in VO2 max or increases in strength with other studies, because heritage wasn't strength-based, but there is around a 50% genetic component. So I think we'll get there. Again, if you're viewing this, how we get there is what I said 10 minutes ago. You can rewind and rewatch. We're going to get there with more advanced sequencing and machine learning algorithms to deal with the data. What other blood markers? I mean, obviously, I think the age-related decrements in sex hormones play a role. There are studies where they've taken out the sex organs in rodents and shown a few things. One of which is, we can see this decrease in musculature. We can see this decreased motivation to perform voluntary exercise. So that's an extreme model, but when you go through andropause as a guy my age, it's just tougher to get to the gym and it's harder to adapt. And I think some of that is testosterone driven. If we supplement back to levels when I was 25, I'm going to see an enhanced response. So I think the blood work serves value there. There's newer types of analyses that can be done. They will be expensive. One thing we're doing right now in collaboration with Lim Taylor at Mary Hardin Baylor is we are looking at nanoparticles in the blood. They're called EVs, and they carry microRNA cargo. And we're interested in looking at the molecular signature between trained female, untrained female, and then what happens after one bout of resistance exercise, and then whether that acute response relates back to some of the phenotype there in terms of muscularity according to DEXA, et cetera, et cetera. So, some of that may be informative. In addition, our colleague and dear friend Chris Van, right, he's working with large data sets from the STRIDE study at Duke, seeing if there's predictive value with looking at some circulating EVs, microRNAs, and whether or not that is related to phenotype. And he is using regression models, machine learning, and I think you're going to see a combination of these nanoparticles in the blood being somewhat predictive. So, yeah, there's certainly things moving forward. Looking back at this podcast 10, 20 years, I think some of these things will come to fruition. The big question is, can we commercialize it? Can they be available to us, whether we, order it online and do an at-home test and mail it back in, or come to a place like Fitomics and do it? I think, I think we'll get there. So,
Hey, I received that. Man, what are you in closing?
Yeah.
What are you most excited about with? I know you just mentioned a handful of things that you're working on right now.
Sure.
There's a lot of, what would you say, volatility in the academic. We'll call it the academy globally right now.
Yeah,
with
sure.
funding, etc. It's a little bit tumultuous out there.
It is.
You have creatively, and I think with high integrity, leveraged industry relationships.
Yeah.
To help support, because this stuff is expensive.
Yeah, it's
to
not
do.
cheap.
Yeah.
Yeah.
What are you most excited about in the next 3 to 5 years, as far as what you're actively working on, the direction of the molecular and applied science lab?
So, I was never a person to develop a 3 to 5 year plan. And I learned that from Frank Booth. I am in the moment, immersed in what we're doing, and we pivot, right? Case in point, sarcoplasmic hypertrophy, right?
Right.
We pivoted, and that led to some productive publications and us learning and us getting to collaborate with Troy and Stu and others. And I just can't speak enough to the ingenuity of the students and what they do and how that drives my passion. And so examples would be Max Michel with his disuse study, right? He looked at people that came into the study with resistance training experience, people that came into the study who didn't train. We put a leg brace on them for 2 weeks. We saw atrophy, similar magnitude, by the way, whether you were trained or not. That's a mind blow. And then we recovery resisted trained them for 8 weeks. And as a matter of fact, when you talk about relative hypertrophy, those that were untrained gained a higher relative amount of muscle mass, more so than those that had prior training. So, and now we've done transcriptomics, proteomics. We're trying to explain that. but that's going to spin off into probably other leg bracing models, or wearing a boot, and getting calf biopsies, or not even doing biopsies. Danny, another student in the lab, Danny Plotkin, he's working with the MRI Center right now because he did that with his length and contraction study. I think that's fascinating. Depending upon what we see there, that may spin off. But in addition to that, with the MRI engineers, we're hoping to get what's called DTI imaging, which can look at muscle volume and fascicle orientation. And so that would just be super cool. And I'd probably invest a lot of time in terms of how aging, how exercise modality affects certain outcomes there. And then in addition to that, right, Brad Rupel, he did a study where we leveraged deep proteomics from a company called Sear. We sent our samples to Sear, and we got so many targets in that muscle tissue. It was an 8-week training paradigm where it was unilateral leg training, pre-post intervention, deep proteomics, and we saw that. Interestingly, resistance training didn't really alter the proteomic profile. So that was interesting because we said, well, okay, is that only in older individuals? So now that's leading us to ideas doing an age comparison, right? Do we have this sort of proteomic lack of plasticity with aging with resistance training, at least with that model? Maybe more training helps, speaking to the responsiveness versus a young population. And in addition to that, that deep proteomics enabled more insight than we had prior knowledge of, meaning that regardless of training, we also had some young muscle in that analysis, but it looked like really cool mechanisms are affected when you talk about muscle aging, one of which is alternative splicing of transcripts before they're exported from the nucleus. So there are a lot of proteins in muscle in older individuals that would lead to more alternative splicing, which then probably leads to the accumulation of proteins that aren't functional in muscle, right? And as a matter of fact, Ben Miller, he's at OMRF. I just recently went to a conference and he talked about the accumulation of misfolded and non-functional proteins with aging. So I'm like, okay, this is great, right? Like I really can get excited about investigating this. And then what can we do about that?
Right. Yeah.
My guess would be resistance training probably is a safeguard against that phenomenon, but to be determined. We got to do that study. In addition to that, another mechanism that we saw with our deep proteomics in older versus younger individuals, regardless of training the older individuals, was proteins in relation to the export of the mRNAs. They're impaired. And so now with aging, we're sort of painting a more complete picture of, we have obviously the loss of muscle mass. We know that if you don't exercise, you're going to lose it. If you exercise and do the adequate intensity, volume, modality, etc., you can prolong the age at which you eventually fall off the cliff, which we think is probably around 80. But I'm very interested in those molecular mechanisms, and it looks like, based on our deep proteomics, what could be happening is this myonuclear instability. That we have more splicing leading to more misfolded or non-functional protein accumulation. In addition to that, we're not getting transcripts out of the nucleus as effectively. And then you combine that with what we already know from the literature. Older people have less satellite cells, in particular satellite cells that are associated with type 2 myofibers. Now, if you train, you can reverse that, right? But now we have a really nice picture, which seems to indicate, at the end of the day, keep resistance training and do it correctly.
Yeah, that's a good conclusion, man. Hey, fascinating stuff. That's exciting.
Thank you.
We're really honored to have you here. I could ramble on about how thankful I am for you, but thank you.
I'm thankful for you too, sir.
Yeah, thank you for everything.
Thank you, Dr. Lawson.
Yeah.
David, thank you so
Hey,
much.
Muscle for life, man. We love you. Thank you.
Yeah. Thank you, guys. And I look forward, hopefully, doing this again soon.
All right. Peace out.